Here's a riddle. I'm invisible to the eye, yet found in every cell. I hold instructions for life, but can't do the work myself. What am I? That's right, DNA. From bananas to humans, this twisting code stores life's blueprint. But the mystery is, how does that code transform into proteins, the true workers of the cell? Let's find out. In this video, you'll learn how DNA becomes RNA, how RNA becomes protein, and why this process is known as the central dogma of biology. Inside the cell's nucleus, DNA acts as the master blueprint. It stores instructions for making proteins, the building blocks of life. Meanwhile, a gene is a segment of DNA with instructions for one specific protein. Each gene tells the cell what kind of protein to make and when to make it. Taken together, this flow of information is called the central dogma of molecular biology. DNA makes RNA and RNA makes protein. So, what's the first step? It's transcription. An enzyme called RNA polymerase reads the DNA code and builds a matching strand of messenger RNA, or mRNA. Think of mRNA as a temporary copy of a gene. It carries the DNA instructions from the nucleus to the ribosome, the protein factory. Next comes translation. The ribosome reads the mRNA like a barcode, turning the genetic code into a chain of amino acids, the building blocks of proteins. Here's how. The ribosome reads 3 letters at a time, called codons, and matches them with tRNA molecules. Each tRNA carries a specific amino acid that fits the code. As the ribosome moves along, amino acids are added one by one, linking together into a polypeptide chain. This chain then folds into a specific protein with a unique job. And here's the key transition. After translation, the protein is folded and sometimes modified before it actually gets to work, whether that's building tissue, digesting food, or sending signals. We've just followed the entire journey, from DNA code to a folded protein ready for action. Want to turn those milestones into mastery? Tap the link to access the practice test, flashcards, and study guide, then jump right back into the video. Lock in what you learned and keep going. Now, with that foundation in place, let's see why accuracy is so critical. If even one codon is wrong, the entire protein might fold incorrectly, which can cause diseases or malfunctions. For example, in sickle cell disease, just one mutation in the gene for hemoglobin changes a single amino acid. That tiny change makes the protein fold incorrectly, causing red blood cells to take on a sickle shape and leading to serious health problems. Mutations like this show how a single change in DNA can sometimes create harmful proteins, while others may have little to no effect. Additionally, proteins themselves perform thousands of jobs. They're enzymes, hormones, receptors, and even muscle fibers. They make life possible. In fact, every function in your body, from blinking to breathing, depends on protein synthesis, following the DNA instructions inside each cell. So let's recap. Transcription happens in the nucleus to make mRNA. Translation happens in the cytoplasm at the ribosome to make protein. Together, these steps explain the central dogma of molecular biology. Ready to quiz yourself with the content covered in this video? Check out the link in the description where you'll find Quizlet content made just for you.